Three of them are really which are of interest. Are one really that is very close to our heart is we really want to request you to join our feedback program, because your ideas and your feedback is critically important in building and Reaxys to what it is today and what we want it to be in the future. We want to build Reaxys for chemists, and this we can only do by working very closely with you. Second thing in the downloads item, you'll see our contact details of the Reaxys team, so you can reach out to them directly and ask them questions. Also, for students, if they are here, or not even students, there is Boehringer Ingelheim's, join the team, and please look at, Boehringer Ingelheim is actively recruiting, so please look at the download section for further details. With this, I would like to introduce our next speakers. Our next speakers, and importantly, before I forget this, I've been reminded about this a couple of times. Please remember that the presentations will be available for on-demand view for the next 6 weeks. So in case you want to revisit it again, please note that these recorded presentations are available on demand for the next 6 weeks on the same platform. With this, I would like to introduce our next speakers. Greg actually mentioned Swiss Biosphere, so I'm very happy to introduce Vincent. I'll jump to the interesting part, which is before I talk about his PhD and etc. But very happy to say Vincent is one of the significant contributors, if I must say. Although he is quite modest about this, but he is one of the most significant contributors to building the whole family of Swiss things for medicinal chemists. Swiss docking, SwissTargetPrediction, where we collaborated quite closely, Swiss Biosphere, Swiss ADME. So we are very happy to have Vincent here. He has a PhD in organic chemistry from University of Lille. He is an associate professor in molecular modeling in the Department of Oncology, and he joined a molecular modeling group in the Swiss Institute of Bioinformatics at Lausanne, where he is currently also the group leader together with Olivier Michelin. So thank you, Vincent, for joining. And along with Vincent, we have Aurora. She also has a PhD in organic chemistry. She has and focuses a lot on cheminformatics. So we have got two really good cheminformatics experts on this talk. It is going to be really interesting. Aurora really supports Elsevier's clients to utilize the rich data sets that we have to help build AI and machine learning based models, or they don't necessarily need to be AI and machine learning based, any kind of model, to be honest. To answer the question. She has over 12 years of experience in data management and analysis software solutions for pharma and biotech industries. And we are happy to have both Vincent and Aurora. With this, I hand it over to Vincent.
Thank you very much for the introduction. Thank you also for the kind invitation. It's a real pleasure to be here. And I would like also to thank Greg for mentioning Swiss Biosphere some minutes ago. Yeah, so I will take this talk as an occasion to present you the SwissTargetPrediction tool that we have developed in the lab in order to predict the protein targets of bioactive small molecules. And beyond that, I would like also to show how it was possible for us to perform a large-scale evaluation of this tool thanks to the data coming from Reaxys. So, predicting the protein targets of bioactive compounds can be seen a bit as counterintuitive at first, because obviously we designed the small molecule to be active against a primary target, and we could think that this relationship is explaining the bioactivity of the compound. But in fact, the situation is a bit more complicated. First, because on average, bioactive compounds have several possible secondary targets. We generally think that a drug has about 8 different targets. And this is what we know. It could be more, actually. And reversely, for some of the bioactive compounds, and even for some of the drugs, we don't know what the target is. So we know that they are bioactive, but we don't know the mode of action. And so there is a need to predict those primary and secondary targets. And the secondary targets being possibly negative, like creating some toxicity, or positive and responsible for polypharmacology or drug repurposing. There is really a need to achieve this and answer this question. So in the laboratory, we have decided to tackle this question using an artificial intelligence approach. And here I would just like to make a short aparté about this, because there might be some confusion in the public about artificial intelligence and deep learning due to the recent emergence of software like ChatGPT. So I would just like to define exactly where SwissTargetPrediction is situated in this ecosystem. So artificial intelligence actually has a very wide definition. Basically, it's every system, every software that is able to take a decision or make a prediction by mimicking the human intelligence. It can be a very simple program that tells your vacuum cleaner robot to turn when there is an obstacle in front of it, or it can be a good old chase simulator that calculates all the possible moves before taking a decision. So these simple tasks actually are already considered artificial intelligence. Within artificial intelligence, we find a subclass which is called machine learning, which basically uses the existing data and imitates the way a human being would learn from them in order to take those decisions and make those predictions. Actually, within this field, we find the typical quantitative structure-activity relationship, QSAR, and actually medicinal chemistry was among the very first fields in science to implement a machine learning. That was in the 1960s, basically. And within machine learning, we find yet another subclass, which is deep learning, which uses multi-layer neural network in order to extract information for data and perform the different tasks. And this is in this category that we find ChatGPT, Midjourney, and AlphaFold. So you can get that these approaches can be extremely efficient, but they required a lot of data for the training. The training can be tricky itself, and often, not always, but often, they can be also difficult in order to interpret the results from a deep learning. They can act as a black box. And this can be a bit difficult in a drug design project where the confidence between the modelers and the medicinal chemist is very important. If I predict the target of a protein, of a small molecule, but I cannot explain why I make this prediction, this could break actually the confidence. So basically, we decided to learn a machine learning approach and to stay with this statistical learning in order to be able to be more interpretable in the results. So basically, in SwissTargetPrediction, we are using the similarity principle, which says that if two molecules are very similar, they are prone to be active on the same target. And this is really artificial intelligence because it's totally mimic what we are doing. If I need to predict myself as a human being what could be the target of the bioactive small molecule, I would just think about all the molecules in my mind that looks like this small compound, remind what the targets are for those compounds, and this way make a short list of possible targets that could come to my mind. And we are going to do exactly that thing with SwissTargetPrediction. To better understand this similarity principle, I'm taking an example here, which is the structure of the kinase domain of BRAF that I have cut in order to show and display the binding site. And in this binding site, I have superimposed different kinase inhibitors. Now, if I extract them and keep them in their bioactive conformation, we see that they all share a common shape, actually, which is a kind of wave that you better see with the two upper compounds, actually. And this shape similarity between the compound is explained by the fact that for a strong binding, these molecules need to be complementary to the shape of the binding site, and we can see that on the screen. And the molecule needs also to display hydrogen bond donors and acceptors at the same position, non-polar function at the same position, so on and so forth. And so this is a source of the shape complementarity, just the fact to be complementary to the binding site of the protein. This also translates into the chemical structure. So basically you see here that all those compounds, all those BRAF inhibitors have similar chemical moieties in similar position in their structure. So aromatic rings, aliphatic, nonpolar rings, hydrogen bond donors and acceptors, all at the same position because they need to be complementary to the molecular interaction that the protein could do with the moieties in the different position of the binding site. So again, the complementarity of the compounds that they need to have in order to be strong binders of BRAF translate into similarities of chemical structures. So now that we know that we need to seek for chemical structure similarity or shape similarity, we need also to quantify those concepts. This is very important for machine learning, basically. So when we need to compare two molecules and determine and give a value to their similarity, what we do is to use what we call fingerprints. So I don't have the time now to explain the technology behind the scene, but basically we transform the chemical structure of a molecule into a vector, and the content of the vector depends on the presence or the absence of chemical moieties into the molecule. So now if we want to compare two chemical structures, we can compare their fingerprints, their corresponding vectors, and we get a score that ranges from 0 for totally different molecules to 1 for identical compounds. And so this way we can quantify the chemical similarity of the two compounds. We can do the same for the shape. So here we will transform the shape into a vector that is going to encode the shape of the molecule in the 3D space, but also the way those molecules are projecting electrostatic charges and atomic contributions to the lipophilicity. Again, comparing the shape of the two molecules will boil down then to comparing the two vectors, and we will get a value between 0 and 1, 0 for totally different shapes and 1 for identical shapes. So now that we can transform molecules and the molecular similarity into values, and we can quantify the similarity principle, it is possible to obtain that kind of curves here that we obtained some years ago using molecules that we picked randomly from Campbell. You see here the blue curve corresponding to the probability that two molecules taken randomly actually share the same target or one target as a function of their shape similarity. And we see that above a threshold of about 0.90, so 90% shape similarity, we start to be able to predict that indeed the two molecules could share a common target. So capitalizing on this, it's possible to make the prediction. So basically, in our case, a prediction will correspond to a reverse screening. So if we start from a compound, a query compound for which we would like to determine what could be the potential targets, we are going to look into a large library of compounds for which we know the target, which ones are similar to this query compound. And from this information, we will be able to infer a list of possible targets together with some probability of the query compound to be active on those targets. So to do this, we perform this way in SwissTargetPrediction. So we started from Campbell, which is basically the academic little brother of the medicinal chemistry part of Reaxys. So don't worry, I will come back to Reaxys just a bit later, but we started from Campbell then. We extracted all the drug-like molecules that were found experimentally within a binding assay to be active against a set of human proteins. And we extracted this way 400,000 different compounds that are active on more than 2,000 human proteins. Once we had those compounds in hand, we transform them into the chemical structure and the shape-based vectors, so the fingerprints, so that we could easily compare them, their structure and their shape, to a query compound. So the second step was to encode the chemical structure and the shape into vectors, and now we can perform the reverse screening. So we take the query compound, we transform it also into vectors corresponding to the chemical structure and the shape, and we compare those vectors to every vectors of all the compounds in our library, and we separated those compounds per target, so that this way we can determine which molecule that is a ligand of target A is the closest one in terms of chemical structure. And this is giving us what we call the 2D score. We can make the same for the shape, looking for the most similar ligand of target A in terms of shape to the query molecule, and this will give us the 3D score. What we did for target A, we can do it for target B, C, D, and all the targets of these 2000 sets that we discussed a bit earlier. And when this is done, when we have the 2D score and the 3D score of our compound for all the possible targets, we fill that into a logistic regression that we train again from the Campbell data. And this logistic regression allows us to calculate the probability with which the bioactive compound is going to bind to target B, A, Z, so on and so forth. So we can rank them based on the probability, and we can provide the user with the top 1, 5 of 15 most probable target among a list of 2000. So unfortunately, I don't have the time to explain how we train this logistic regression in detail, but you can find this information in the literature in our papers. So once we have trained this, this model, we tested its robustness using cross-validation, but we also tested its predictive ability using a small external test set of molecules that were not in Campbell at the time of the training. And for 72% of them, we were able to predict at least one of the experimentally known targets within the top 15 predicted target among 2000 possibilities. So this was quite encouraging, and we decided to release this tool in the form of a website that we are going to discuss a bit later. But before that, I would like to show why we decided to use two definitions of similarity, one based on the chemical structures as every other competitors, and one based on the shape this time. So basically, you see here an exercise. We know that the query compound is a ligand of the calcium channel, and now we would like to predict that. So what we do at first when we predict it is to reverse screen the ligands of all the targets, including the calcium channel. And when we reverse the ligand of the calcium channel, we found that the closest molecule in terms of chemical structure is the one that you see now on the screen. And this molecule is so similar to the query compound that it is in fact the most similar among the 400,000 molecules in our dataset, and this allows us to rank this target as the most probable one for the query compound. But obviously, this is actually not really a challenging prediction. It's totally obvious. The two molecules differ by only one hydroxyl function, so no big deal, but still this can be very useful because we cannot know by heart 400,000 molecules along with their target. So if we can retrieve that in a couple of seconds, like we have seen within the Reaxys interface together with Greg, then this is already very useful, and this is why our competitors are using this type of approach. But what happens when the molecule is very new? It's the first one in its chemical class for a given target. This is another exercise corresponding to that. So again, this is an exercise. We know that this query compound is a CDK1 inhibitor, and we want to predict it. So at first, we are going to make the reverse screen, and when we reverse screen the ligand of CDK1, we find that the closest molecule in terms of chemical structure is this one. But it's quite different. It's only around 700 among 400,000, so not very good. And if we only consider this, the target is going to be ranked 168 among 2,000, so it would be a bad prediction, basically. Now, if we look for the closest one in terms of shape and projection of charges and lipophilicity in space, then we find that the closest molecule and CDK1 ligand is actually this compound, which is the third closest one among the 400,000 compounds. And this allows us to rank this target in number 5 among 2,000. And number 5 is actually enough in order to convince someone to make the essays and test this possible target among maybe the top 10 or top 15. So this is actually a useful prediction. And so we see that using the shape similarity is actually complementing the chemical structure similarity and is very helpful when the compounds are totally new in terms of chemistry. So we put this approach behind a web server that is called SwissTargetPrediction.ch. You can just draw the molecule or provide it in the SMILES notation. And after 15 to 20 seconds, we can provide a list of possible targets along some probability that actually, if the molecule is bioactive, then it would bind on this particular target. So I told you that we actually tested the predictive ability of the approach on 500 molecules that was available to us at the time. And so there is always, it's always a bit frightening actually to release such a tool to the worldwide community, because we are not absolutely sure that the predictive ability will be the one expected and will be very useful for the scientific community. So one way to see that is to see the engagement of the users. So if the users are using the tool and come back and publish the results of the tools, then we can figure out that this is actually useful. And this is what we do. Actually, we follow what the users are doing on our websites. And you see the increase in the number of jobs that are submitted every year on SwissTargetPrediction. So this year we should reach about 1.7 million jobs submitted on SwissTargetPrediction. And you see also that the number of citations of this tool is increasing. We should reach about 800 citations this year too. So you can see that there is an attrition rate between the number of jobs that are being done and the number of citations. And this is quite explainable actually. So first of all, we made a survey, and we know that about 45% of the users are actually students at the bachelor and the master level. And obviously they are doing jobs on SwissTargetPrediction for educational purposes. And so they will not publish their results. Also, when we publish a research paper in medicinal chemistry, for instance, in GMedChem, we know that we are not just publishing one molecule. We are going to publish tens, hundreds of molecules that were synthesized and tested experimentally. And beyond this series, there are hundreds, maybe thousands of molecules that were considered virtually and that were tested using in silico approaches such as SwissTargetPrediction. So that's why actually one paper can correspond to hundreds or even thousands jobs for different tools like SwissTargetPredictions and others. So thanks to Reaxys, actually, it was possible now to make a very large and unprecedentedly large, actually, test of such an approach to really decipher the exact prediction ability of this reverse screening technology. So basically, thanks to the fact that the medicinal chemistry part of Reaxys was put at our disposal, it was possible to extract a part of it, and this part corresponds to all the drug-like molecules with the same definition we used previously that are active on a binding assay against a protein, a human protein. And the set is so large at the beginning that even after filtering, we find 364,000 molecules that not only are bioactive on human proteins, but that were not present in the Campbell database at the time. So there are new external molecules on top of the ones that we use for the reverse screening and the training of the test. So this is a huge amount of data that was available. And on top of the pure number also, what was interesting for us was the chemical diversity. Actually, about half of the scaffold of the molecules that were coming from Reaxys were not present in Campbell and vice versa. So basically, there was a possibility to test the approaches not only on classical molecules that we could find in Campbell, but also on new ones that are not in the training set of the approach. So basically, another thing that was very important for us for some technical reasons that I'm not going to explore right now is the fact that we need to identify unambiguously the target of every single bioactive molecule, and not only the target, but also the target class. And this was made possible at that time thanks to the new Reaxys taxonomy that was made available to us some years ago. So this taxonomy was very efficient, and thanks to this, we had absolutely all the data in hand to easily perform the test set and the benchmark of our approach. And here are the results. So basically, if we take those 360,000 compounds and make the prediction of their possible targets, Even if we only consider the top ranked target among 2001, we were right 51% of the cases, which is very encouraging. And if we consider the top 15 possible targets, then on average, we could find one experimental target in 73%. So 73% is very close to the 72% that we had some years ago when we tested that on a small set of 500 molecules. But here the statistics are much more robust because we tested that on nearly 1,000 more molecules here. And thanks to this wealth of data from Reaxys, it was possible to study the success rate beyond just the raw data. So here what you see in this graph is the evolution of the predictive ability of our tool at different ranks as a function of the number of ligands that are known to be active on a given target. And we see that for targets where you have more than 1,000 ligands, the success rate at rank 15 can reach up to 80% instead of 72. And you see here the dependencies that we have to this number of ligands per target. And actually, this is quite intuitive. The more ligand you have, the better the comparison can be, the more robust it can be. But for the first time, thanks to this amount of data, it was possible to quantify this effect that you see here. Another effect that we considered was not only the amount of molecule, but also the diversity of the molecule, which is very important because if you have 2,000 molecules, but they are all based on the same scaffold. At the end of the day, you just have a very limited information. And so we investigated that thanks to the Reaxys data, and we find out that if you have, again, more than 500 scaffolds known to bind to a given target, then you can increase the success rate again to about 80% at rank 15. You can also reach about 75% when you have 100 to 500 scaffold, which is actually quite frequent, as we can see on the left here graph. So this was very important to quantify this. Again, this is totally intuitive, but if you can put figures behind that, then you know you can better actually inform the users about the quality of the predictions that you make because you know how many ligands you have for a given target and how many scaffolds you have for a given target in the reverse screening. Yet another question that we could address thanks to the Reaxys data was actually going back to this question that we investigated with the two exercises before. So what is going to happen for this prediction if the compound is totally new and is not absolutely within the chemical class of the known ligands of the target? We could address this because of the diversity and the large scale of the Reaxys dataset. Here, we only considered the compounds that have absolutely no scaffold in common with the compound of the training set, excluding them using two very large definitions of scaffolds, so Morco and Aurora. So it was a very harsh filtering, and only about 10% of the molecules are able to pass through this filtering. But since we had 360,000 compounds at our disposal, 10% is already 32,000 different compounds, and this allows to make robust statistics. And indeed, you see here the impact that we have on the predictive ability when your compound does not look anything at all like a molecule of the training set. So obviously there is an impact. The question is much more difficult in this case, but the impact on the success rate is more limited. So it's about 50% less success rate at rank 1 and 10% success rate less at rank 15. This is still very comfortable if you need to make a prediction, basically. So this is true for SwissTargetPrediction, but beyond that, it would be true for whatever other approach that would be based on this reverse screening and molecular similarity principle. So you can guess that since we trained the model on Campbell and tested it on Reaxys, we had the idea of doing the reverse and training the model on the Reaxys data set and potentially test it using the Campbell data. So we did it actually in collaboration with the Reaxys team. And I will not go into the details, but one interesting finding that we have is that the coefficient of the logistic regressions that we have to calculate the probabilities that we introduced rapidly some slides ago, actually you see here their evolution as a function of the number of heavy atoms in the correct compound. And you see that the curves that we trained from Reaxys are nearly identical to those that we trained from Campbell. And this is really reassuring because the physics behind the scenes, so the molecular interaction between a ligand and a protein, remains totally universal. It does not depend on the database. And finding exactly the same model at the end of the day actually was really meaningful for us. And again, this can only be investigated if you have access to two very large and quite different data sets at the end of the day. But what we can also observe is that the curves that we obtain for Reaxys are more robust, they are more consistent, which means that the coefficients are less noisy. And this was due to the fact that we had a larger number of active molecules for the given targets that we investigated. On top of that, we have also a larger chemical diversity in Reaxys, which is favorable for the prediction as we have seen earlier. But this is beyond this presentation, unfortunately, so maybe another time or in a paper we could investigate more in detail this question. So it will be all for me today. I would like to thank the members of the Computer-Aided Molecular Engineering Group of the University of Lausanne, of the Molecular Modeling Group of the Swiss Institute of Bioinformatics, and in particular Antoine Daina, who is very active in the design and the development of SwissTargetPrediction, and who also made all the statistics that I showed today. I would also like to thank Reaxys for putting all this information at our disposal, in particular Peter Kadeff, Olivier Barberan, and Rosaline Sanke. And with this, I will be happy to take your question in a couple of minutes from now. Thank you very much.
Great. And I think we need to thank you so much for this work, Vincent. It's and to also greet everyone. Say hello, and thank you for taking the time to join today the Global Research User Day and being with us. My name is Aurora Costache. I am a Customer Engagement Manager from the Customer Success team. And today in this talk, I would like to briefly introduce you to a different way to access Reaxys content via data as a service, and use it at large, use the content at large to basically build suitable and impactful work like Vincent just presented, to predict models and so forth. And also I'd like to introduce you to our team, the customer success team, which I'm part of, and explain a little bit the relationship you'll have with us when you will basically acquire the data from Reaxys in order to build, predictive models and so forth. Bioemphasize is one of the success or many success engagement stories, building or co-creating a data set for predicting a blood-brain barrier model. So this broad and deep curated chemistry content, which is available in Reaxys, now can be accessed not just via web interface, but also through this new solution of data as a service, where you can access it at large via API, so application programming interfaces, and basically have access to this experimental validated chemistry data from literature, so you can use it for predictive models, analytics, and so forth. Depending on the need and also of the delivery to the full database, but also to data subsets, and the reactions data, where basically you have access to the reaction chemical database, and the substances which are coming with their properties as well. And last but not least, the bioactivity data, where you have access to the targets and the bioactivity which are reported for these targets in literature. And basically this off-platform data, which comes in a variety of formats, are the files for the reactions data, so you can easily integrate it with your existing data. SD for substances, XML formats, and JSON formats, for the rest of the data. This could be applied, at large scale for data mining and processes, and use it on a variety of use cases. And here are just a few use cases. You can use the data as large as presented here in this slide. So you can use the Reaxys data integrated with your own data to build predictive models, to build predictive properties, do QSL in silico screening. You can even integrate your own Reaxys data. You can use the data to build internal dashboards for visualization or custom reports, do federated searches, or use, for example, Reaxys data to improve your synthesis optimization as well. And I'll touch briefly in this how we use Reaxys data to build a data set to build a predictive model to see if the compounds are passing the break-bad barrier. Reaxys data sets are already recognized in data science projects by peer industry, and they're published in Science, for example. Here it's an example how Jensen Group used the Reaxys data to basically enhance and design their processes. And moving on the other side, you can see how AstraZeneca uses integrated Reaxys data to improve their predictive algorithms and basically develop and enhance the drug processes. Also, on the bottom left, it's an exemplification how Mark Porter's lab uses Reaxys data to build the predictive retrosynthesis algorithm, and that proudly sits now into the .com solutions. And I hope you had the chance to join the morning session to hear this story. And in the middle here is presented how Samsung used the Reaxys data to build their own retrosynthesis module and deployed internally. Last but not least, it's an example here how on the bottom right corner, how Novartis group of researchers use the Reaxys bioactivity and targets data to build this GitHub library, chemogenetic library, where data curated or bioactivity data curated from multiple sources were created in a database, AI ready, which will basically give a tremendous help to the bio community to build models. But these success stories, many success stories, unfortunately are shadowed by a lot of failure stories. And in fact, there are studies that more than 85% of the data science projects are actually failing. And the main reason of that is because the emphasis is still on the technology, on the models, and so forth, and less on the data, on understanding the data, on the curating the data, and basically the complexity of it, and what sits and be fitted in this technology to actually have a successful model, a successful outcome. And this is basically what we observe when we interact with our customers as well. And that's why we decided to add this highly consultative process on top of just delivering the data and hand it to the customer, where basically we sit with the customer and make them understand the complexity of the data, how to manage it, how to control it, how to be successful when they actually use it in building these models. And just to explain very quickly the way we work with the data, because as you can imagine, it could be quite complex. When you focus on the delivery, on an ingestion, so more on the technical side, we come in and basically focus on not the technology itself, but on the users, the ones which will consume the data, and work with them to understand the data structure, the data complexity, to efficiently use it for the project they have in mind, and also to discover new projects. So our team, the customer success team, which I'm part of and very proud of, really has the goal in mind to enable scientists like you to understand the data, to use the data at the full potential by providing education, training, consultancy, and also advising on the change management, because all this digitization process, it's not just about the technology, but it's about the users itself. And us being certified professionals, process certified professionals, we can advise and also increase the usability and decrease the basically the not the usage of the data. And to wrap up the presentation today, I would like to emphasize one of the success examples and how we realize that this type of consultancy is really needed and beneficial when working with our customers and they work with our data. So basically, here is an example of a data set put together for building a blood-brain barrier model to a large pharma company or large organization, where basically they had the Reaxys data ingested in their data lake. And one of the team there decided that they can use their internal data with the Reaxys data to put together a data set to build an in vivo KP, brain blood barrier prediction model. At that time, the team wasn't aware that the data can be accessed via data as a service or via API and FAT files. And once they discovered that and they tried to do, then that by the way very successful. So they didn't use the data at the full potential. Process seems to be very simple. You retrieve the information or the relevant parameter for that particular model you would like to build. The verification process, it's quite laborious, and I quickly explained that to have a data set which could be AI ready to train the model and to do the predictions. And basically, to prepare this data set to be AI ready involved data wrangling, basically to normalize the data, to remove the improper data, to fill the missing information, and also to enhance it scientifically and curate the data on that respect as well. And I explain in the next slide what I'm trying to say about that. And all this process, which is the most laborious when you prepare data sets for building models like Vincent described, it's something you can add more, thoughts into it. So the outcome at the end, when the data set was created, basically in comparison, and we compare more than 40 public domain available data sets, was that the data set created with the data from Reaxys basically was larger in quantity. So it was the largest data set by far, but also was, in terms of quality, was better because we were able to cover, and this is where the scientific, consultancy was added. So we were able to cover factors which were relevant to values beyond just the structure itself, like the dose, the administration route, the species, the detection time, and so forth. And all of that basically really showed that when we did a benchmark, that the chemical space was far larger, and the chemical diversity, as Vincent mentioned for his model as well, was larger compared with the largest public model available out there. And that was also reflected on the accuracy of the predictions of the model itself. So with that, in very short presentation, I was asked to present today. Really hope that I convince you that Reaxys dataset are recognized and used and add significant value in a data science project, in data analytics, and beyond that, that when you acquire and work with data from us, from Reaxys, You are provided with scientific support and navigation of the data sets of the data itself of the computer. If you want to hear more about other use cases and how to start us, please feel free to contact us. With that, I would like to really thank you for your attention, and I am happy to answer any questions.
Perfect. Thank you very much, Vincent and sorry. Thank you very much, both of you, for this great talk. So there are a couple of interesting questions. So let me put this first. Vincent, quite a lot for you. First and foremost, I guess all the pharmaceutical company people would like to know if the searches on Swiss Biosphere or Swiss Target Prediction are proprietary. Where are they stored? What's security? Can they be retrieved? Can they be searched by anyone?
Yeah. So the results are available under the CC BY 4 license, which means that they are free to be used by whoever submitted the calculation, including pharmaceutical companies. This is a requirement actually from the Swiss Institute of Bioinformatics those days. Regarding the security of the data, what we do is that we delete regularly all the submissions that have been done on the website. We don't look at the molecules that have been submitted on the website ourselves, so we keep that totally anonymous, and we don't have a look at the molecules. Of course, it's a public website. This means that if a very gifted hacker is attacking the website, we cannot be absolutely sure that the data is safe. So this is the reason why we regularly delete the data, to be sure that if something is broken, we will just have a very little leak, if I can say so, but we cannot be more secure than that, basically.
Thank you very much. There's another question from Christopher. He says that Campbell includes a prediction on its compounds, but it is not that good. Have you checked the difference between their and your prediction?
No, we have not made a benchmark against the competition. So one problem is that, to be fair, we need to do a benchmark on a large amount of data and a diverse amount of data. So otherwise, we could be the first or the last one just by chance. And if we want to do that, then this means that we need to have exactly the good version of the software directly on our machines and not through a website, because if we need to submit tens of thousands of compounds through a website, it's going to be endless. So yeah, we don't really have the time to make full benchmarks like that. Basically, what we do is a comparison of the quality of the approach indirectly by just having a look at the number of citation jobs that are submitted and so on, hoping that if you are cited a lot, then you should be good.
Thank you, Vincent. Last question, because we are running over time. Ahmed's question: can any vector be used for compounds when targeting a protein? Can you perhaps comment on that, Vincent? Can any vectors be used?
Sorry, I didn't know it was for me. I'm not sure to understand exactly the question. So basically, the model does not depend on the protein. It only depends on the similarity principle and the similarity between the correct compound and the ligands of known targets. So we do not tweak the model for a given protein. This could potentially be the case. You have models like PASS actually that make a model per target, but yeah, it's not the case in Swiss Target Prediction.
All right. With that, I would like to thank both of you, Aurora and Vincent, for a very engaging conversation. There are some questions still unanswered. We will get back to you shortly, but we have our next speakers, very exciting talk ahead of us.